What is a genetic disease? A genetic disease is an inherited medical condition caused by a DNA abnormality. This can include any missing, extra, or irregular portions of chromosomal DNA. It can arise from an odd number of chromosomes or unusual structure of one or more chromosomes. An example of genetic disease would be polycystic kidney disease or PKD.
During interphase, this is how the cell looks when it first forms. Here is chromosome 1 and 2 located in the nuclei, each with a chromosome from mom and dad. The centrosome is located in the cytosol of the cell and is composed of mainly protein. The cell grows and goes through synthesis, which is the duplication of DNA. The centrosome duplicates as well, and the cell's nuclear membrane begins to disintegrate. In prophase 1, the homologous pairs come together and wrap around each other tightly. This is the point where they are switching segments of their chromosomes, which is also known as recombination. Crossing over with non-sister chromatids is what occurs when pieces of genetic material is traded with homologous chromosomes. This is important because the chromosomes that came from mom and dad will be passed on to the child as a brand new chromosome, which maintains variability. Recombination ensures that the next generation will be different than the current one. Now in metaphase 1, the homologous pairs line up along the metaphase plate. The centrosomes move to either side of the cell, and the spindle fibers attach to the centromere of each chromosome. Independent orientation is a position in which the chromosome pairs assort themselves. Looking at two pairs of chromosomes, there are 4 different possible combinations in which a chromosome can orient itself. Now looking at the 23 pairs, there are over 8 million ways these chromosomes could independently orient themselves. Along with recombination and crossing over, this is another factor that increases the genetic variability of an organism. In anaphase 1, the chromosomes are being pulled apart and shipped to either side of the cell. The homologous chromosomes are now separated. In telophase 1, a nuclei is being formed around each set of chromosomes, and cytokinesis splits the cell in half. During prophase 2, crossing over is no longer required, so the chromosomes line up along the middle of the cells in the metaphase plate. The spindle fibers of the centrosomes now attach to the centromeres once again. During anaphase 2, each of those chromosomes are being pulled apart to either side of the cells. In telophase 2, nuclei are being formed around each of the pair of chromosomes, and cytokinesis splits the cells into 4, now forming 4 new. For the genetics of polycystic kidney disease, it affects chromosomes 4, 6, or 16. These are the 3 affecting proteins that cause polycystic kidney disease. What are the symptoms of polycystic kidney disease? The symptoms include high blood pressure, back or side pain, headache, kidney failure, blood in urine, and frequent urination. To treat high blood pressure, diet should include low sodium and low fat, and exercise should be increased as well as a decrease of stress. For pain, if it's moderate, over-the-counter medication would be prescribed with acetaminophen, and if severe, surgery may be required to remove cysts. For blood in the urine, lots of fluid should be intaked in order to dilute the urine, and for kidney failure, treatment may include dialysis or kidney transplant.
Mantle cell lymphoma results from malignant transformation of a B lymphocyte in the outer edge of a lymph node follicle. The transformed B lymphocyte grows in an uncontrolled way, resulting in the accumulation of lymphoma cells, which causes enlargement of lymph nodes. In the G2 phase of interphase, a nuclear envelope encloses the nucleus. The cell grows as it completes preparation for cell division. In prophase, the nuclear envelope disappears. The mitotic spindles begin to form, and the duplicated chromosomes appear as 2 identical sister chromatids. In metaphase, microtubules extend from each centromere across the cell, and chromatids of each duplicated chromosome connect with the mitotic spindle at their newly formed kinetochores. In metaphase, the centromeres are now at the opposite poles of the cell, and the chromosomes have lined up along the metaphase plate. In anaphase, the two daughter chromosomes begin moving toward opposite ends of the cell as their kinetochore microtubules shorten. In telophase and cytokinesis, the poles are pulling away from each other, forming the cleavage furrow. The two daughter cells form shortly after the end of mitosis. The cause of MCL is due to cyclin D1 on chromosomes 11 and 14. Cyclin D1 is overexpressed, causing a large accumulation of MCL cells. Density-dependent inhibition is a cell that stops dividing in a space once it is in contact with another cell. And anchorage dependence is when a cell is in contact with a surface to divide on. Essentially, cancer lacks these qualities. There are 4 stages to cancer. Stage 1, lymph node region or single organ is affected. Stage 2, 2 or more lymph node regions are on the same side of the diaphragm are affected. Stage 3, 2 or more lymph nodes on above and below the diaphragm are affected. Stage 4, widespread disease in lymph nodes or in any other body parts are affected. Treatment for MCL is a problem due to no curative treatments, with chemotherapy as a main form of treatment. And with difficult treatment, only a poor prognosis of roughly 18 months or 2 to 3 years of survival can be given.